Hello class, this is BIOT643, Techniques of Biotechnology, and this is a supplementary review lecture on the fundamentals of molecular biology. I've created this lecture to help you review some of the basic concepts in biochemistry and molecular biology that will help you be successful as we discuss numerous techniques in biotechnology throughout the semester. As an outline for what we'll be covering in this lecture, we will be starting off looking at some of the biological macromolecules, starting off with nucleic acids in general, and then a little bit more specifically on DNA, RNA, and proteins, respectively. Then we will take an even closer look at DNA, starting off with the pairing between the bases, the double helix structure, an in-depth look at DNA melting, and then the structural organization into chromosomes and plasmids. Next, we'll take a look at how all that information encoded in the DNA gets turned into life. And so we'll start off looking at the central dogma. Then we will examine nucleic acid features related to transcription and translation. And then we'll take a look at the three critical processes of replication, transcription, and translation. Now, most of this material you all should have been exposed to previously, either in your undergraduate programs or here at UMUC in BIOT 601, or maybe some of your other classes. But I did want to create this lecture just to help you guys have a review of some of that material, because I do feel that some of the techniques we'll be talking about do hinge on a detailed understanding of some of these. So we're starting off looking at some of the biological macromolecules. These do include nucleic acids and proteins, but they do also include some other molecules such as carbohydrates and fatty acids, but we will not be talking about those here in this lecture. We will be looking only at the nucleic acids and the proteins. Nucleic acids are made up of three basic structural components. The first is a five-sided sugar ring. In DNA, this is deoxyribose, and in RNA, this is ribose. And we'll take a closer look at that in just a little bit. The next component is a phosphate group, and this is similar between DNA and RNA. And the last component is a nitrogenous base. In DNA, we have adenine, thymine, guanine, and cytosine, commonly referred to as A, T, C, and G. And in RNA, we have A, C, and G, but the thymine T does not exist in RNA. It is instead replaced with uracil, commonly abbreviated as U. These components are organized into a structure with a repeating pattern. A nitrogenous base is covalently bound to a sugar ring. Off of that sugar ring is a covalently bound phosphate group. Attached to that phosphate group is an additional sugar ring, also with a nitrogenous base attached. To that sugar ring is attached another phosphate group, which is attached to another sugar, and so on. The sugar determines whether it is DNA or RNA, and you see here on the left deoxyribose, which is the sugar that forms the backbone of DNA, and ribose on the right, which forms the sugar that forms the backbone of RNA. And structurally, these are identical, with the exception of the group that is bound to the 2' carbon, and you can see that the carbons of the ring are numbered 1', 2', 3', 4', and 5', and we'll take a look at some of these in more detail on the next slide. But here you see that in deoxyribose we have only a hydrogen group, whereas in ribose we have an OH, a hydroxyl group, and The difference between these is that the oxygen is missing in deoxyribose, hence the name deoxyribose. Taking a closer look at the numbering scheme that I just mentioned on the previous slide, we have here a nucleotide of deoxyribose, and this is just as an example. The numbering scheme between deoxyribose and ribose is the same. Taking a look first at the 1' carbon, this is where the nitrogenous base is bound. The 2' carbon is the one that has a different group attached to it, whether it is DNA or RNA. The 3' carbon has a hydroxyl group, and when we look at polymerization in the upcoming slides, we'll see that this particular hydroxyl group is critical in order to form the DNA or RNA chain. The 4' carbon essentially has nothing particularly special about it, except that it has the 5' carbon attached to it. The 5' carbon is not actually part of the ring. As you can see, it is bound to and above the 4' carbon, but we still number that as the 5' carbon. The 5' carbon has the phosphate group attached to it, and together with the hydroxyl group on the 3' carbon, this particular phosphate group is critical for polymerization, as we'll see. So taking a closer look at DNA specifically, to help us review the structure of a DNA strand, we have here a short single strand of DNA composed of 4 nucleotides. The top one has a T attached to it. It is followed by a phosphodiester bond, after which there's another sugar ring with an A attached to it, followed by another phosphodiester bond and a sugar ring with a C, and another phosphodiester bond with a sugar with a G attached to it. So you can sort of think of the 5' carbon as the top of any particular nucleotide, and the 3' as sort of the bottom of any particular nucleotide. And so you can see here on the top one you have the 3' at the bottom, and the OH group off of that 3' forms a bond with the phosphate group at the 5' of the next nucleotide. And so the electrons on that O of the OH group will bind to the phosphate group on here, and then now those two first nucleotides would be bound together covalently. The OH on the second nucleotide would bind to the phosphate group on the third nucleotide, and then the OH group on the third nucleotide would bind to the phosphate on the fourth nucleotide. And so you can see here that the 5' and 3' top and bottom of the nucleotide stays constant, and so what we have is essentially an overall direction of 5' to 3', as in the top and the bottom of the particular DNA strand. And typically when we write DNA sequence, you see them listed as 5' to 3', and that's where this comes. So now let's take a look at RNA. Here we have an RNA strand on the right, next to a DNA strand, the same figure that we saw at the end of the previous DNA section. And again, you can see that the DNA at the 2' carbon all have an H group bound to it, while the RNA ribose sugar rings all have an OH bound to them. And again, the other difference between DNA and RNA is that DNA contains a thymine, while RNA contains a uracil. There are 3 primary types of RNA. There are additional types that we will discuss during the RNA lecture, but as part of the review of transcription and translation, we'll only discuss these particular three during this lecture. So the first is ribosomal RNA, or rRNA. That forms the structural scaffold of the ribosome. The second is mRNA, or messenger RNA. That is the molecule that is created during transcription and carries the information from the DNA to the ribosome to be translated. The last is transfer RNA, or tRNA, and this is part of the translation process by which the information on the mRNA is directly translated into protein. And the tRNA has two active sites. One is the anticodon, which recognizes the 3 base pair sequence on the mRNA, and that's known as the codon. And the second site is the aminoacyl site that has a specific amino acid attached to it. And that amino acid that is attached corresponds to the codon, and that amino acid gets bound to the previous amino acid, hence creating the protein chain. This diagram is a good overview of the various roles that RNA plays. On the left, you see in yellow the DNA strand. Up at the top, transcribed off of the DNA, are molecules of rRNA. These form the structural scaffold of the ribosomes, and so these rRNA molecules form complexes with the ribosomal proteins, creating 2 ribosomal subunits, which then combine to actually carry out the process of translation. In the middle, off of the DNA molecule, we see transcribed the mRNA. That carries the information from the DNA over to the ribosomes, which then translate it into the protein with the help of the tRNAs, which are also transcribed from the DNA. And you see the amino acid at the bottom end of the tRNA molecules. And so mRNA is involved in the process of translation, actually in 3 different respects. Here we see some diagrams showing the structure of the ribosomes. On the left we have the prokaryotic ribosome, and on the right the eukaryotic ribosome. They are comprised of 2 different subunits, as I mentioned earlier. They are different sizes, and so they are creatively referred to as the large subunit and the small subunit. On the left, we see the prokaryotic large subunit is comprised of 2 different rRNA molecules, referred to as the 23S and the 5S, and these fold up to form a structural scaffold for 31 different proteins. And so together, those components, the 2 RNA molecules and the proteins, make up the large subunit. The small subunit of the prokaryotes is made up of 16S rRNA and 21 different proteins. And then you can see the eukaryotic small subunit is made up of 18S RNA and 33 proteins. And the large subunit is made up of 3 different rRNA molecules together with 46 proteins. mRNA is the molecule that carries the genetic information encoded in the DNA to the ribosome for translation. The gene that is encoded on the DNA is transcribed into mRNA, which is a temporary RNA copy of the information encoded on the gene. The information encoded on the mRNA molecule is organized into 3 base pair sections called codons. Each codon encodes for a single amino acid. On the example sequence in the figure on the right, you can see that the first 3 base pairs, AUG, encode the amino acid methionine. The next three base pairs, AAA, code for the amino acid lysine. The next, CUU, encode leucine, and so on. The transfer RNA, or tRNA, is a single strand of RNA with nucleotides at various regions that are complementary to nucleotides at other regions along the molecule. These regions, because they are complementary, are able to base pair with each other. If you look at the figure on the right, you can see the crossbars between the strands indicating where the hydrogen bonds are. This allows the single strand of RNA to actually form in various sections a double-stranded complex between two different parts of the same strand of RNA. This allows the overall molecule to take on a three-dimensional shape. As you can see in both of these figures, the one on the left is more detailed, the one on the right is more cartoony, but is probably indicates the structure overall a little bit better. Down at the bottom on the anticodon loop, you have the three nucleotides that form the anticodon, which will recognize the codon. We'll see that on the next slide. And up at the top, you see the amino acid binding site, which not surprisingly binds the amino acid that will be included on the nascent protein corresponding to the codon. Here we see an even simpler representation of the tRNA molecules. At the top you have the amino acid binding sites, and at the bottom of each you have their respective anticodons. The outgoing empty tRNA has the anticodon ACC, and that was bound previously to the UGG codon on the mRNA, and now that it has already donated its tryptophan amino acid, the tRNA molecule is displaced. The next tRNA molecule has the anticodon UUU, which is bound to AAA on the mRNA, and that encodes lysine. The GAU codon that's next encodes aspartic acid, and the UUC codon coming up in the on the ribosome's path encodes for phenylalanine, and that is recognized by the AAG anticodon on the tRNA. Next, let's take a quick look at proteins. The structure of an amino acid is comprised of 4 different groups that are covalently bound to a central carbon atom. These are the amino group, the carboxyl group, the hydrogen, and a variable group, usually referred to as R, that differs between each amino acid. The R group is what defines the amino acid and differentiates it from all the other amino acids. Here we see the biologically relevant amino acid residues. You can see up at the top right that the amino acid structure overall has simply been reduced to a small brown plane, and coming up off of that is the R group. So the first one we see, for example, is alanine, which at that central carbon has within that plane the amino group, the hydrogen, and the carboxyl group, and then coming up off of that is its particular residue, R group, which is the CH3 methyl group, and the various other amino acids are shown here. These are simply for completeness to show the various groups. You should generally know these, but you won't be required to know in detail their particular structures or anything. So proteins are amino acid chains, analogous to how we made strands of DNA from individual nucleotides. You can see the at the top part of the figure here the peptide bond formation, and so you have on the left of the equilibrium arrows, two different amino acids, one with residue group R1, one with R2, and you have the amino groups of either of them on the left and the carboxyl groups on the right. Once these combine, you have a peptide bond form between the two amino acids, and then you have a diamino acid chain with the amino group on the left and the carboxyl group on the right. And in the bottom part of the figure 215 on the slide, you can see a chain of 5 different amino acids, R1, R2, R3, R4, and R5, and you see the same overall structure with the amino group on the right of each and the carboxyl on the left. And so, analogous to how we were defining the directionality of nucleic acids from 5' to 3', over in proteins, we have an overall directionality that we refer to as the N-terminus for the amino, because it has a nitrogen, and the C-terminus, which is the carbon from the carboxyl group. And so we write these sequences of proteins from N-terminus to C-terminus, just like we did 5' to 3' for nucleic acids. Here we have another diagram showing a 5 amino acid protein comprised of tyrosine, glycine, phenylalanine, and leucine. And you can see again the directionality from N-terminus to C-terminus. Now let's take a closer look at some important concepts regarding DNA, starting with the pairing between the bases. As I mentioned previously, hydrogen bonds can form between the bases when they're in proximity to each other. The standard or Watson-Crick base pairs are A and T and G and C, each of which bind to each other. As you can see, each of them have at least one electronegative atom, the nitrogens or oxygens. These line up with the respective hydrogens, and so they are able to form hydrogen bonds between the electronegative atoms and the hydrogens on the corresponding base. Here we have double-stranded DNA showing the base pairs hydrogen bonding to each other. On the left, we have a strand of DNA with the 5' up at the top and the 3' end at the bottom. And so you have this particular 5' to 3' directionality. It's important to note that the opposite strand has the opposite directionality, with the 5' end at the bottom and the 3' end at the top. Now let's take a look at how that forms the double helix structure that is the characteristic hallmark of DNA. The term for the opposite directionality that I showed on the previous diagram is anti-parallel. This indicates that one strand is in one orientation and the other is in the opposite orientation. When these strands get long enough, they are able to wrap around a central axis, and they form this double helix that is characteristic. There's approximately 10 nucleotides per turn of the helix, and the bases, as we discussed already, are bound to each other, A to T and G to C. Here we see two different representations of the DNA double helix structure, the ball and stick model and the space filling model, and in each of them you can see the minor groove and major groove that correspond to different parts of the DNA helix twist. Now let's take a look at DNA melting, which is a very important topic to understand for some of the technologies that we'll be talking about throughout the semester, starting with PCR in Lecture 1. The important thing to remember about hydrogen bonds is that, unlike covalent bonds, they're weak. This means that two molecules that are held together by hydrogen bonds can have those bonds broken. In the case of the DNA helix, we refer to this as denaturing or melting. It is a reversible process in that if you bring the strands back together in the proper orientation, the hydrogen bonds between the same bases can reform the same base pairs, and the DNA two strands will once again form the double helix structure. This reversible denaturing is critical to DNA replication and other natural processes that occur within the cells. And in vitro, we use this for various processes, starting with PCR. And so the two strands come apart when the hydrogen bonds are broken, which does require an energy input to break those bonds. Biologically, within our cells, this is typically mediated by ATP, which provides chemical energy in order to break those bonds. And in the laboratory, typically during PCR, we will use primarily thermal energy in order to break those bonds. Here again, we see our short strand of double-stranded DNA, anti-parallel with hydrogen bonds between the bases on either strand. Up here on the diagram on the top right, it's important to remember that the number of hydrogen bonds is inversely proportional to the amount of energy within the system. And so in this diagram, we see on the left a nice orderly system with a lot of hydrogen bonds holding the molecules together in an orderly lattice structure. As you increase the amount of energy, that structure gets increasingly disordered as the number of hydrogen bonds is decreasing, and ultimately becomes a completely disordered system of high energy with very few hydrogen bonds that are stable formed. And so we can think of this process in terms of water, for example, in which the hydrogen from one molecule will form a hydrogen bond with the oxygen from a different water molecule. And so at low temperature or low energy state, this would form hydrogen bonds between all the various molecules and would result in an orderly solid lattice of, in this case, ice. And as we were to increase the amount of energy, for example, thermal energy, in the system, we would end up with eventually liquid water and then ultimately steam as the number of hydrogen bonds between the various molecules decreased as a result of the increasing energy. So we can do the same denaturing with DNA by applying energy in order to break the bonds that hold together the two different anti-parallel strands. The amount of energy required to break the hydrogen bonds holding the two individual bases together varies based on the base pair. As you can see in the A and T base pair, there are two hydrogen bonds holding the two bases together. This bond strength requires an energy of 5 kilocalories per mole to break, whereas in the GC base pair, there are 3 hydrogen bonds holding the bases together, and this requires a bond energy of 6.9 kilocalories per mole. The specific numbers are not terribly important to us, except for the fact that you should know that a GC base pair requires much more energy than an AT base pair to break. Those varying bond strengths have implications on the amount of energy it takes to separate the two strands that comprise a specific DNA sequence. We refer to the temperature at which these two strands separate as the DNA melting temperature, or TM. we can calculate the Tm using a fairly easy formula of 2 degrees Celsius times the number of A and T base pairs plus 4 degrees Celsius plus the number of G and C base pairs. So for example, here we have a string of 20 AT bases, 5' to 3', and the Tm for this would be 2 degrees Celsius times 20 nucleotides of A and T plus 4 degrees Celsius times 0 because there are no GC bases. And so 20 times 2 is 40. So that would give us a Tm for this particular sequence of 40 degrees Celsius. On the flip side, we have a sequence comprised of solely GCs. And so we would have 2 degrees times 0 plus 4 degrees times 20 nucleotides. And we would end up with 80 for a Tm. And you can see that in an evenly mixed sequence of A, T, C's, and G's, you would have 10 bases of A and T, so 10 times 2 is 20, plus the 10 base pairs of G and C, so 10 times 4 is 40. 40 plus 20 gives us a Tm for this sequence of 60. Now this equation is fairly easy to use. It's not terribly accurate. It is good for short sequences, but obviously, for example, if you had a 100 base pair sequence, you would end up with a Tm of several hundred, which is certainly not accurate. So this is only accurate for short sequences. But what it does allow us to do is understand the contribution that each of these nucleotides have to the amount of energy it takes. So as we discussed, the GC bases require more energy, and this formula shows that fairly well. It's important to realize that melting does not occur instantly at the Tm. The melting temperature is the temperature at which 50% of a DNA sequence has been denatured. If we plot that on a graph of temperature versus the single-stranded DNA molecule percentage, we see a sigmoidal curve where at low temperatures there is 0% or essentially 0% of molecules are single-stranded, which means that essentially 100% of them are double-stranded. So melting has not occurred. They have not been denatured. At the right end of the graph, we see at high temperature that nearly 100% of the molecules are in their single-stranded formation and therefore have been denatured. The Tm, as I said, is the temperature at which 50% of these molecules are single-stranded or double-stranded, because the other 50% are clearly double-stranded. And so the Tm is defined at this point where 50% are double-stranded. And so in this particular graph, we have a Tm of 60 degrees Celsius. So taking a closer look at this graph, as I said, at the low end of the graph, we have essentially all the DNA molecules in their double-stranded formation. At the Tm, we have half of them, 50%, in their single-stranded formation. And at the higher end, we have nearly all the DNA molecules in their single-stranded formations. It is important to realize that statistically, when you're talking about so many molecules, even at very low temperatures, you will have some of the DNA molecules which are in their single-stranded formations, and similarly at very high temperatures, you'll always have just a few DNA molecules which are in their double-stranded formation. And so This graph asymptotically approaches 0 and 100% at either end, but you can never actually achieve 0 or 100%. But for the purposes that we're discussing, we essentially refer to them as 0 and 100% within some statistical error margin. So we just looked at some of the extremes on the graph, at the low end, at the high end, and at exactly 50%. Let's look at some spots in between. If we were to pick this spot here along the graph, we would be looking at approximately 10%. This appears to be about 57 degrees or so. Whereas if we look up here at about 63 degrees, we're looking at about 95% of molecules in their single-stranded formation. So it's important to realize that this DNA melting curve is a continuum of a large number of molecules, the percentage of which is increasing from approximately 0% to approximately 100% as the temperature increases, and the TM is right in the middle, where 50% of them are in their single-stranded formation. Here we see the DNA melting curve that we were discussing previously in blue, with its TM at 60 degrees. And we also have on this same graph a second DNA melting curve shown in green. This represents a sequence that is more AT rich, and so it has more AT base pairs than does the sequence shown on the graph in blue. What this results in is an overall shift to the left of the graph. The curve maintains its sigmoidal shape. The parameters that we discussed as far as the various points at which a certain number of molecules are in their single-stranded formation, all of this is the same, just shifted down to a lower temperature. So here we can see that the TM of this particular curve is about 56 degrees, and so at a lower temperature, 4 degrees lower than the other graph, is where 50% of its strands are in their single-stranded formation. Now we've added an additional DNA melting curve, this one shown in red. This represents a more GC-rich DNA sequence. As you can see, the overall shift of the curve relative to the blue curve is to the right, toward higher temperatures. And we can see that the TM is much higher than it was in the blue curve. Here the TM is approximately 64 degrees. And so at 64 degrees would be where 50% of the red curves molecules are in their single-stranded formation. So now let's take a brief look at the organization of DNA into chromosomes and plasmids. Here we're discussing primarily bacterial chromosomes. Obviously, the eukaryotic genome is organized into very different types of chromosomes. The purpose of this lecture is to lead directly into some of the introductory material that we'll see in the first couple weeks, primarily cloning into bacteria. So I set up this slide to focus specifically on the bacterial chromosomes and not the eukaryotic chromosomes. I will be discussing those a little bit later in the semester, but for now we're just looking at the prokaryotic chromosomes. So within bacteria, there is a wide variety in the chromosomes that we see in different genera and species. Typically, they have one chromosome, circular in shape. There are variations of that. Some bacteria have multiple chromosomes. Some of them have linear chromosomes. And so there's no hard and fast rules that are always adhered to. But generally, you do have a single circular chromosome that accounts for the majority of the organism's DNA. And so, for example, in the bacteria E. coli, that particular chromosome is about 4.6 megabase pairs. That's million base pairs, and extends a length of about 1.2 millimeters. Bacterial plasmids do not occur in all bacteria. There are many species that do not have plasmids, however many do. Often they are found in multiple copies of the same plasmid within an individual bacterial cell, and oftentimes there will also be various different plasmids within a cell. Typically these are circular. You can sort of think of a plasmid as almost a mini chromosome, very small in size. Often they do encode genes. Usually these genes confer some sort of selective advantage, such as antibiotic resistance to the bacterium that is harboring the plasmid. Some species do have linear plasmids, but there aren't too many of those. And in the electron micrograph here at the bottom of the slide, we actually see a plasmid, PBR322. This plasmid is about 4.3 kilobases in length. It is circular, and it forms the basis of a lot of commercial vectors that you can order for your research. They are based on this particular plasmid. If you recall, I mentioned that the E. coli genome is approximately 1.2 millimeters in length. That's actually really huge when we're talking about the size scale of an individual bacterial cell. So how exactly does all that DNA fit into a tiny microscopic cell? Part of that has to do with the supercoiling of DNA structures. So we see here in the diagram circular chromosomal DNA. There are DNA binding proteins, such as histones and histone-like proteins, that will bind to the DNA and create these loop domains, and that helps sort of compress the overall structure of the DNA into a more compact structure. And then supercoiling will even further compact those DNA structures. So now we see the individual loops on the second part of the diagram get twisted up and sort of fold in on themselves, and so it takes up much less space within the cell this way. And this does occur with both chromosomes and plasmids, and supercoiling is something that we'll have to consider when we talk about gel electrophoresis in lecture 2. So now we've talked about the information that is encoded within the DNA. We've talked some about the role of RNA in transmitting that information. Now let's go ahead and start putting all the pieces together and talk about how we turn that information into life. And we'll start off reviewing the central dogma. The central dogma describes the overall biological information flow necessary to life. We start with information encoded on genes in the chromosomal DNA. The gene is the basic unit of genetic information. DNA undergoes DNA replication, in which copies are made of the existing DNA strands. This is mediated by an enzyme called DNA polymerase. Transcription is the production of an RNA copy of a gene onto messenger RNA. This is mediated by the enzyme RNA polymerase. Translation is when a polypeptide or protein is made from the mRNA by combining amino acids, as we saw on the previous slides. And this is mediated by the ribosome. We will be discussing each of these processes more in depth in the upcoming slides. One important point to mention on the previous slide is that I intentionally left off reverse transcription. We will be discussing that process in later lectures, but for the sake of reviewing the three basic processes of replication, transcription, and translation, I wanted to avoid overcomplicating things, and so I left that off. But no doubt some of you noticed that that was not on there, and I guarantee I did not forget about reverse transcription, and I will be getting to that in the near future. So now let's look at the nucleic acid features related to transcription and translation. So here we have a diagram of a strand of DNA. There is a gene that is shown in blue that encodes a protein coding sequence, as most genes do. And there are a variety of features located on the nucleic acid strand, both upstream and downstream from the gene coding sequence. And please note that these are not drawn to scale. And so the features related to transcription are shown in green. Those related to translation are shown in orange, and the gene and protein coding sequence are shown in blue. So first we have furthest 5' upstream various regulatory sequences. Then we have the promoter. That tends to be the main driver of how many RNA transcripts are produced of the gene. Regulatory sequences also play some role in that, though to a lesser extent, but a non-negligible extent. Next up, we have the transcription start site. That is where the transcription actually starts, not surprisingly from the name. So the RNA polymerase will bind to the promoter, and then it recognizes a sequence on the promoter. Once it has bound, it will start to move down the DNA strand until it recognizes the transcription start site, and then at that point it will start producing an mRNA molecule that's complementary to the DNA of this strand. So the next feature that's related to transcription, all the way at the 3' end, is called the terminator, and we will be discussing all of these in more detail shortly. So once transcription occurs, as I mentioned, it starts at the transcription start site. So the mRNA copy that is made does not include the promoter, the regulatory sequences, or any of the DNA sequence upstream of the transcription start site. The ribosome binding site, which was encoded on the DNA but is not active on the DNA, it does not become an actual entity itself until it is encoded on the mRNA. That is not surprisingly where the ribosome will bind. It will continue moving down the mRNA molecule until it finds the start codon. Typically this is the AUG that we saw earlier. That will prompt the ribosome to start assembling the polypeptide, and then it will stop assembling the polypeptide once it hits the stop codon. Translation occurs, and we see the polypeptide now, the protein, made of various amino acids, and we see the N to C terminus directionality. Typically, the first amino acid is methionine, which is encoded by AUG. This slide simply describes in text the features that I just described on the previous slide. I put this here so you'd have an easier time reading these particular descriptions, so read this at your leisure. Now let's take a look at DNA replication, which is making a DNA copy of an existing strand of DNA. This occurs by semi-conservative DNA replication. It's referred to as semi-conservative because you have a double-stranded DNA molecule, and at replication, the two strands will separate at what is called the replication fork. The original strands are shown in dark blue. The new strands, or the daughter strands, are shown being synthesized in a light blue. They are synthesized by the complementarity of the base pairs, but what we end up with is at the end two different DNA molecules, one that had the actual strand that was in the original molecule bound to a newly synthesized strand, and the other second DNA molecule that's produced has the same makeup, one of the original strands and one newly synthesized strand. And you can see this here in this diagram as well, in more of a flat format. As I mentioned, the newly synthesized strand is formed based on the complementarity of the base pairs. So if there's one thing I want you to take away from our discussion here on DNA replication, it is that every strand of DNA sequence inherently contains the instructions to make a copy of itself. So if we imagine that this arrow is a DNA molecule, a single strand, we can see the 5' and 3' directionality, and that's which way the arrow is pointing. This strand, of course, would be a long sequence of nucleotides. For the purposes of our diagram, we're just looking at these 9 in the middle. So we have GGA, TCC, ACG. This is a single strand, so it would be bound to the other complementary strand. The sequence on this strand, because we're reading always from 5' to 3', the sequence of this strand is CGT, GGA, TCC. As you can see, that can be a little bit hard to do sometimes, but it is fairly straightforward. So when DNA replication occurs, the bonds between the two strands will break, and they will separate. Here I'm showing them completely separate. In reality, we would have the replication fork that we saw on the previous slide, and at the relevant section where replication is occurring, they would be separate, but much like a half unzipped zipper, part of the molecule that is not being replicated would still be bound together. But for now, let's talk about them as though they were entirely separate, just for simplicity. So now we have these two different strands that are complementary to each other, perfectly exactly bind together, but now we have separated them. When we make a DNA copy of this particular strand with the DNA polymerase, it forms a complementary copy of each nucleotide and binds that. So now we see again CGT on the top strand, GGA and TCC, because those are the complementary base pairs to the sequence shown on the original blue strand. And here we have the complementary base pairs to the original green strand, and those there we have GGA, TCC, ACG, and as it turns out, these two are exactly identical copies of each other. So the blue copy has the instructions to make a green copy, which in turn has the instructions to make a blue copy, because you have this complementarity between the base pairs. You can always make the sort of opposite copy of the DNA strand, and because of this we can continue separating out these two new strands and make 4 molecules if we were to fill in the strands of those, and this process continues, and every time our cells divide or bacterial cells divide, this is what's occurring. An entirely new copy of the genome is made and given to each new cell. DNA polymerase requires a primer to begin the polymerization reaction. In vivo, a short RNA primer is created by an enzyme called primase. The RNA primer that it creates is complementary to the DNA sequence at that site. Then the DNA polymerase comes, and from the end of that RNA primer will synthesize the DNA downstream in the complementary base pair manner to the DNA template. The RNA primer is then degraded and replaced by a DNA sequence. The reason DNA polymerase requires a primer is because the enzyme needs a nucleotide to already be present on the strand that is being synthesized in order to form a phosphodiester bond connecting that nucleotide to the next one that is being added. Here in the diagram we see the template strand, and on the strand that is being synthesized opposite it we see the nucleotide that is complementary. You see the bases hydrogen bonded there at the top. Here on that nucleotide you see the OH group attached to the 3' carbon. As was mentioned previously, that OH group is critical to the polymerization reaction, as we'll see here in this slide. Down here we see the base of the next nucleotide that will be added. So this base is further up 5' on the template strand. If you notice, the 5' to 3' directionality is pointing up the slide. However, that base is 3' if we're using the reference of the newly synthesized strand's directionality. So the complementary nucleotide to that base will come along, start to be incorporated into the strand, the newly synthesized strand. The hydrogen bond is formed based on the standard base pairing rules. So that hydrogen bond forms, but that's that weak hydrogen bond that we talked about. This nucleotide is not part of the newly synthesized strand yet, so it is a DNTP. We see that it's a deoxyribonucleotide. So we see the base on there, we see the sugar ring, and we see this triphosphate group. The triphosphate group is not incorporated into the strand. We only have a single phosphate group. So somehow we need to get rid of two of the three phosphate groups that we have here. So what happens in the polymerization reaction is that the electrons from the OH on the previous nucleotide will attack the phosphorus atom here on the first of the phosphate groups. That will cause the diphosphate, also referred to as inorganic phosphate, to be released, and that will form a phosphodiester bond. And now we have that new nucleotide that we added as part of the newly synthesized strand. And then down here at the bottom, we'll be adding the next nucleotide in exactly the same manner as we saw in this slide. And so you see on the newly synthesized strand, we have 5' at the top, we have 3' at the bottom, and so the direction of strand synthesis is from 5' to 3'. DNA polymerase only builds in that direction. It actually cannot build in the 3' to 5' direction. So now that we've reviewed replication, let's move on to transcription, which is the creation of an RNA copy of a DNA sequence, usually one that encodes a protein. So before we actually start talking about the components of transcription, it's important to point out that many of these slides, including the current section here on transcription, are taken primarily from the mechanisms and processes used by prokaryotic organisms. The reason for focusing on the prokaryotes is because generally the eukaryotic mechanisms and components and overall processes are far more complicated. For example, with transcription, there's a whole slew of eukaryotic transcription factors that are involved in the process. And for the purposes of reviewing the basic concepts of, in this case, transcription, I felt it best to focus primarily on the simpler version of the model of the system, in this case the prokaryotes, and sort of gloss over the eukaryotic systems, but later in the semester, as necessary, I'd be happy to bring these up again, look at the eukaryotic variant of the system, and talk about how it's different from what we may have learned earlier in the semester, etc. But in terms of refresher, I wanted to focus on the simpler version of the system to kind of help jog people's memory or reintroduce the subject if it's been a while since you've looked at it. So with that said, we'll take a look at the components of transcription in prokaryotes. As I mentioned earlier on the nucleic acid feature slide, the gene that is to be expressed is that transcription is regulated by an upstream promoter. Of course, upstream meaning 5' to the gene. And then RNA polymerase holoenzyme is the, we generally simply refer to it as RNA polymerase, but that is the overall enzyme that is made of multiple subunits that come together, as we saw on the, no, we haven't seen that yet, excuse me. And so all these components come together and are able to produce an mRNA copy of the gene that is going to be expressed. Now, one of these subunits, the sigma subunit, is the one that recognizes the promoter. There are different types of sigma subunits. The nomenclature differs based on the organism, but for example, there's sigma A, sigma B, sigma H. Each of these recognize a different promoter sequence, and we'll be looking at some of the promoter sequences in the upcoming slides. But for example, a sigma A promoter has a certain sequence that is recognized by the sigma A subunit, and thereby an RNA polymerase enzyme that has the sigma A subunit, but that promoter would not be recognized, at least not with any efficiency, by an enzyme that has the sigma B subunit. So they are fairly specific. Transcription occurs in 3 stages. These are referred to as initiation, elongation, and termination. We will be discussing each of these individually in more detail in the upcoming slides. But briefly, initiation is where the RNA polymerase recognizes and binds to the promoter and starts building the mRNA strand that will be produced by adding the first nucleotides, ribonucleotides, that are complementary to the sequence that's being transcribed. Elongation, not surprisingly, is where the mRNA copy that has started being made during initiation gets elongated. So the RNA polymerase physically moves down the DNA sequence that's being transcribed, and it continues adding the complementary ribonucleotides. Termination is where the process stops, and the mRNA that's been produced detaches from the RNA polymerase. This is mediated by a structure called a stem loop, which we will look at in the upcoming slides. So we've mentioned several times throughout this lecture a feature of the nucleic acid called the promoter. Now let's take a closer look at exactly what that promoter is, keeping in mind that we're discussing the prokaryotic version of the system. The promoter is the region that's recognized by the RNA polymerase, specifically 2 regions that are recognized by the sigma subunit. These 2 regions, for example, are shown in the diagram. They are centered at 10 and 35 base pairs upstream of the transcription start site. So you see in the diagram the AT that is labeled +1. That is the number nomenclature that's used to indicate that that is the first nucleotide that will be transcribed. Obviously the one after that would be +2, the one after that +3, and so on. If we start counting back the other way, so upstream in the 5' direction, the nucleotide before the transcription start site, which is +1, would be labeled -1. There is no 0, which can be a little bit confusing, but -1, the nucleotide before that is -2, and so on. And so the -10 sequence, here we see that it is TATAAT. I'm reading on the top strand. That sequence is centered at approximately -10. It can go forward or backward just a little bit, but if it moves too much, then it starts to not be recognized. And so the center being approximately -10, that would mean that that middle T or middle A would most likely be exactly -10 up above the transcription start site. Then there is typically another sequence in between that is variable and really doesn't have too much significance, at least not as far as transcription goes. and then the -35 sequence is not surprisingly centered at approximately -35. So 35 base pairs upstream of the transcription start site. And here we see in the example that sequence is TTGACA. And again, one of those middle nucleotides most likely would be right around -35. During transcription initiation, as I mentioned, the RNA polymerase binds to the promoter. You can see the promoter shown here in this bluish-greenish color. The DNA double helix partially unwinds, exposing the bases of the template strand at the site that is to be transcribed. The RNA polymerase begins building the mRNA strand by placing ribonucleotides that are complementary to the exposed DNA sequence, and linking those together with the phosphodiester bonds that we discussed earlier. The RNA polymerase then physically moves down the DNA strand, opening up the strand or the double helix in the direction of the movement to expose more of the DNA sequence to be transcribed, and as it moves down, continues adding nucleotides to that that open section, and we refer to that as a transcription bubble. As you can see on the diagram, the hydrogen bonds between the two strands of the DNA upstream of the transcription bubble are still intact. So it is in its double helix formation. The same is true of the base pairs downstream of the transcription bubble. It's only this section here that locally unwinds and opens up in order to allow the RNA polymerase access to the nucleotides at this site in order to start forming the mRNA. And you can see that molecule in red here. The next step is chain elongation. During this stage, the sigma subunit dissociates. As you recall, the sigma subunit is directly responsible for recognizing the promoter. Once that promoter has been recognized and the transcription process initiated, the sigma subunit is no longer needed, and so it detaches from the RNA polymerase holoenzyme, and the remainder of the polymerase keeps moving down the DNA strand, adding ribonucleotides to the growing transcript of mRNA, thereby elongating it. So we can indirectly visualize the process of transcription occurring using electron microscopy. The process of transcription occurs in parallel with multiple RNA polymerases transcribing a gene at the same time. So as one polymerase enters the elongation stage and moves down the DNA strand, a new polymerase binds to the promoter and initiates transcription. Then as that polymerase enters elongation and moves down the DNA strand, yet another polymerase binds and initiates transcription. Because each of these polymerases are in a different stage of transcription, the length of the mRNA transcript they have produced is different. Additionally, the process of translation also occurs simultaneously to transcription. Because of this, the ribosome, which is responsible for translation, binds to the mRNA transcript as it is being produced by the RNA polymerase. Because the ribosomes bind to the mRNA along their length, we can use these ribosomes to essentially monitor the length of the mRNA. So you can see in the electron micrograph here, we have the DNA strand annotated, The mRNA is being produced by the RNA polymerase, which we can't exactly see. They are small, as is the mRNA strand, since it is just a single strand of nucleic acid. It's certainly harder to see than the DNA strand. But what we can see are the ribosomes, because they are much larger. And so you see moving from left to right, mRNA transcripts. You can see them indirectly by the number of ribosomes that are bound to them. And so the promoter would be off to the left, probably off the electron micrograph, and the gene that is being transcribed, the polymerases that are furthest to the left in the picture are the ones that have just bound and have just started to produce mRNA transcripts. The ones that are further along with the longer mRNA transcripts have been transcribing the DNA sequence for a much longer time and length, and therefore their mRNA transcripts are much longer, giving the ribosomes more space to actually bind on there. And you can see in the diagram on the right, it's essentially the cartoon version of what we see in the electron micrograph. Ribosomes transcribing different length mRNA transcripts, which translates into, well, I shouldn't use the word translates, which manifests itself in different numbers of ribosomes binding, and I find this picture really neat, that we can actually indirectly visualize the mRNA by using the ribosomes. It is important to note this process only occurs in prokaryotes. As you no doubt remember, eukaryotes have a nucleus, and so the process of transcription occurs in the nucleus, and then the mRNAs are produced and then transported out of the nucleus into the cytoplasm, and that is where the ribosomes of a eukaryotic organism are located. So there's this segregation of the processes of translation and transcription, and so this indirect visualization via the ribosomes is something that could not occur, because the mRNA transcripts that would be transported out into the cytoplasm would be complete, and so you wouldn't get this differential length in the way that we could monitor this. So we talked about initiation, we discussed elongation. Now let's take a look at termination of transcription. As I mentioned previously, termination occurs when a stem loop in the mRNA that's being produced causes the RNA polymerase to pause its movement down the DNA strand and its addition of more nucleotides to the nascent mRNA strand. So here in the figure you can see the transcription bubble within the RNA polymerase, and so the two DNA strands are separated. Where the new nucleotides, the ribonucleotides, are being added, we have a temporary hybrid of RNA and DNA. Typically we talk about DNA being double-stranded, so bound to DNA, and RNA we typically discuss as single-stranded, and it generally is. But during the process of transcription, we have this hybrid RNA, DNA bonded together, and so it's sort of a double strand, and it doesn't actually form the double helix that we normally associate with a double-stranded DNA, but it does form this hybrid. The problem with the hybrid is that it's not nearly as stable as, for example, a DNA binding. So what happens when the RNA polymerase pauses is that there's not enough binding strength to hold that together, and so it detaches from the polymerase, and then the polymerase itself detaches from the DNA strand, and that's how termination occurs. So let's take a closer look at the stem loop itself. So you can see here in the left part of the diagram the two regions referred to as inverted repeats. As the name would imply, they are repeat regions, so the sequence between them is the same, but they are inverted relative to each other. So if you see on the inverted repeat on the left, the top strand is AAA, GGC, TCC. That is the same sequence that is on the lower strand. Again, we're reading it 5' to 3', and therefore the complementary sequence is on the upper strand. And so if you mentally eliminate that bottom strand, you can see that if you were to fold that top strand in on itself, the outermost nucleotides would be A and T, which would bind to each other, another A and T, another A and T, then a G and a C, and so what we end up with is the figure on the right, and of course this is mRNA, so we're substituting U's for all of those T's, but you see down at the bottom of this sort of hairpin, it's referred to as the stem loop, down at the bottom you have AU, 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 GC, and so these inverted repeats are direct repeats of each other in the opposite orientation, such that when you fold it in on itself like this, you have binding, and that structure is what causes the RNA polymerase to pause and then eventually detach from the DNA and cause the mRNA to detach from the polymerase as well. Now that we've discussed replication and transcription, let's take a look at the third of the three critical processes, translation, which is the creation of a protein molecule from a strand of mRNA. So in the RNA section, we looked at the general structure of the ribosome. We're going to take a more detailed look at that here in this section. So we saw that it is divided into the large and small subunits. In the bacterial ribosome, these are called the 50S and 30S subunits, respectively. Each of these subunits does consist of mRNA strands, which form the structural scaffold for some additional ribosomal proteins. You can see that here the 50S subunit has the 5S and 23S RNA molecules, along with a number of proteins, and the 30S subunit has the 16S RNA and various proteins. These two combine together to form the 70S ribosome, which is simply the two subunits acting together to form the active ribosome. In eukaryotic ribosomes, these subunits are essentially the same for our purposes, at least. They do have different sedimentation weights, which is where the nomenclature comes from. And so the large subunit is the 60S, the smaller one is the 40S, and the full active ribosome is called the 80S ribosome. So let's take a look, sticking just for, as I mentioned previously, simplicity's sake, at the bacterial ribosome. So let's take a look inside the 70S ribosome. There are 3 active sites for RNA to bind, for tRNA specifically. There is a channel in between the 2 subunits where mRNA feeds through and is read. So the 3 tRNA binding sites are called the E site, P site, and A site, or reading in the direction where they first encounter the mRNA. They would be the A site, which is the aminoacyl, the P site, which is the peptidyl, and the E site, which is the exit. And so you can see here in the B portion of the diagram a little bit more closely how they interact with the mRNA. You can see here at the bottom end of the yellow tRNA that's in the A site, the anticodon of that particular tRNA molecule. It does bind with the bases on the codon. They are complementary, and up at the top of the yellow molecule you see the oval representing the amino acid that's bound there. So looking at the system a little bit more simply, here we see the active ribosome, 50S, 30S, and we're representing the tRNA as just kind of a stick with you see the purple tRNA molecule with an oval up there, again representing the amino acid. So the A site is taken up by this purple tRNA molecule. The P site is the one that is currently being attached, the amino acid is being attached to the growing polypeptide protein molecule, and the blue one in the E site is exiting because it's already had its blue amino acid attached to the polypeptide growing strand, and you see the green P is still attached, the amino acid, to the tRNA. That bond is about to break, and the blue amino acid, which was the first one added in this particular diagram, has already, it's hanging off of the green one there. And so next, after the blue one exits the E site, the green one in the P site currently in this diagram will move into the E site in order to exit the molecule, and the purple one currently in the A site will move over to the P site and attach the green amino acid that's there currently to the purple amino acid, and then you'll have a string of 3 amino acids attached to each other, and then whatever codon is next, that appropriate tRNA will enter the A site, and it just kind of goes like a conveyor belt in there with the tRNAs that have the appropriate amino acids coming in attaching their amino acids in the P site, and then moving out of the molecule through the E site. Here we have essentially the same diagram, although I think this shows the nucleotide interactions a little bit more clearly. We have the P site taken up by a tRNA that is recognizing the codon ACG, and you see the growing protein chain is 5 amino acids long currently. As the ribosome moves, it will attach the next amino acid, which will be the 6th one in the chain. That is the amino acid on the tRNA currently in the A spot. That attachment of that amino acid will occur in the P site once that moves over, and then the tRNA currently on the right of the slide with the anticodon GGA is going to bind to those first 3 nucleotides that currently don't have the tRNA bound to them, the CCU, which are complementary to the anticodon. That will occur in the A site once the ribosome moves over, and again the empty tRNA, once it moves over, that is currently in the P site, will then be in the E site and will exit the ribosome. So that gets us through translation. The process, of course, is a little bit more complex, but we've covered the basics certainly enough to really help understand the techniques that we'll be discussing in this class. So here are the references from this lecture. In addition to this single reference slide, essentially the bibliography, each of these slides in the lecture have the relevant reference from which any of the pictures or general concepts were taken. So I hope you found this introductory lecture 0 useful, and if there are any questions, please feel free to email me, or also post on the discussion on the webpage, because remember, if you have a question, somebody else might have the same question. So I definitely don't mind you guys emailing me directly, but if it's something that you feel you and other students could benefit from, I would encourage you to post on the website rather than emailing me, but I'm very happy to entertain emails. So thank you for listening, and I hope you found it useful.